Lipids are a kind of organic chemical that are extremely important in all biological systems. There are no biological systems that do not depend heavily on lipids in one form or another. Lipids, fats, we're talking about things that are solid at room temperature. This is a product you can purchase. This is called lard. Lard is nothing more than fat that has been taken off an animal and that is used for cooking. Nowadays we know that this is not necessarily the best way to do things, even though sometimes it's quite flavorful. We have oils. Oils are liquid at room temperature. This shows you a bottle of olive oil. It's liquid at room temperature, and olive oil actually can be put into the refrigerator and the freezer and still be liquid because of the type of materials it's made out of. We have fatty acids that we're going to talk about, and fatty acids are long-chain hydrocarbons, which means there's a lot of hydrogen, there's a lot of carbon in it, there's not much oxygen in it. Therefore, it's a different type of molecule than we get with carbohydrates. When we deal with fatty acids, we talk about saturated fatty acids, which have no double bonds, and in this case, we've got some baking sticks, and these baking sticks are basically a hydrogenated vegetable oil. They hydrogenate it to take the double bonds out, which makes it solid at room temperature. We've got the unsaturated ones. We're going back to our bottle of oil, and unsaturated means there are double bonds in there. When we look at these, we will find that the double bonds mean that this particular material is going to be liquid at room temperature. When we break these down, we start looking at fatty acids, which are the basic building block of lipids. The fatty acids, these are common ones that are found. The number of carbons you find in it, the chemical structure of it, the number of double bonds in there, the name, and the melting point. First one, palmitic acid. It says it's got 16 carbons, which means it's going to have 32 hydrogens, and it's going to have 2 oxygens. It's got a melting point of 63 degrees Celsius. Let's put some numbers behind that as well. We're looking for it being liquid at room temperature. Now, the question comes down to it. What's room temperature in Celsius? Well, here's a couple of numbers that help us look at things. 37 degrees Celsius is body temperature, 98.6. So 37 degrees Celsius we know is warm for room temperature. Therefore, 63 degrees Celsius is way beyond 37. So therefore we know this is going to be a fat. Second one down the line says it's got 18 carbons in it. No double bonds. We call that stearic acid. It has got a melting point of 70 degrees Celsius. That's even higher. So the more carbons you put into it, the higher the melting point. We move down one from that and we get oleic acid, which says it has 18 carbons and 1 double bond in it, and all of a sudden we're looking at 16 degrees Celsius. Is 16 degrees Celsius going to be liquid at room temperature? Well, we said 37 is hot, that's body temperature. Another interesting number to remember is 28 degrees Celsius, because 28 degrees Celsius comes out to 82 degrees Fahrenheit. You just flip the numbers around, and 82 degrees Fahrenheit, while that's a little bit warmer than a lot of room temperatures that we'd like to be in, that's a lot warmer than 16 degrees Celsius. So oleic acid is actually going to be liquid at room temperature. The last one down there says it's linoleic acid. It's got 18 carbons. It's got 2 double bonds in it. So this is going to be a polyunsaturated material. Melting point is -5 degrees Celsius. -5 degrees Celsius is going to be colder than the temperature inside of the freezer on top of your refrigerator. So therefore, if you put this material inside of the freezer on your refrigerator, it's still going to be liquid. So the more carbons you have, the higher the melting point. The more degrees of unsaturation you have, the lower the melting point. And all of these, again, are fatty acids, so they are a very unique group in there. We have 2 more words. We have hydrophobic, we have hydrophilic. Hydrophobic means it doesn't like water. Hydrophilic means it likes water. When we talk about fats and oils, they do not like water. If you take a little bit of oil and you put it in with a little bit of water, they don't mix. You shake it up and they mix, and then they will separate. So we have to find a way to be able to move these things around inside of biological systems which are mostly water-based. And one of the ways we do this is using triglycerides. Tri means 3. Glycerol means we're talking about a glycerol molecule. This is a glycerol molecule that's got 3 carbons, it's got 3 oxygens, and it's got 6 hydrogens to it. So it is a carbohydrate. What happens here is we take our fatty acids like this, and you can see that there's a fatty acid, here's another fatty acid. The first one looks like it's got 16 carbons, the second one looks like it's got 18 carbons, and the third one has got 18 carbons in a double bond. These are lined up where the hydroxyl units on the glycerol is near the hydroxyl units on the fatty acids. The process that occurs here is these hydroxyl units come together, water is taken out of this, again it goes back to what we call dehydration synthesis, where water is taken out, you get a linkage between the carbons and an oxygen in the middle of them. The triglyceride, tri meaning 3 fatty acids, and the glycerol. It all gets combined together and there's your molecule. The glycerol part of it is hydrophilic, the lipid part of it is hydrophobic. So we have a molecule like that. Another type of molecule we get is what we call a phospholipid. And a phospholipid has a phosphorus attached to the lipid. Now you can have other things attached to the phosphorus as well. In this particular case, a particular phospholipid has different kinds of fatty acids attached to it. And you could take this phospholipid and you could attach it to carbons or amino acids or all sorts of other things. Phospholipids are important because they become the building block of membranes. And when we look at membranes, and it doesn't matter what type of organism you're talking about, be it a bacterium or a higher animal, they all have phospholipids in the membrane. Another kind of lipid that we have are called sterols. Now these have very complex rings. This shows you a sterol. This would be something like cholesterol. There are a lot of different kinds of sterols because all we have to do is change where the double bonds are and change where the hydroxyl units are, and we go out and we have a lot of different ones. Each little bend in here is where you've got a carbon represented. So rather than making this a really complex formula, what we've done is we've made it somewhat simple like this by just showing you where the carbons are, and on every spot in there you're either going to have a double bond or you're going to have hydrogens in there. So you can see this is rather complex. These sterols are used for things like stabilization of membranes. In human diet we talk about cholesterol, and cholesterol stabilizes membranes, but if you got too much of it, it makes them hard, and that's not a good concept in your circulatory system. Sterols also act as hormones, which means they stimulate reactions, and so they're quite interesting in that nature as well. Another group we have is acute, Suberin, and wax, and these are long chain polymerized hydrocarbons. And when we say long chain, you take a 16 carbon unit and you put 2 of them together and you're going to get 32 carbons. You take a 16 and an 18, you're going to get 34, and you take a 34 and a 16 and all of a sudden we're up to around 50 carbons. And so it makes these things absolutely long, huge long chains in there. Difference is where they work at and what they do. If we look at this, all we're doing is taking these, we're putting them together, and by putting them together we get these rather long units in there. When you look at the surface of this leaf, what you see is this red material on the outside of it, and that is cutin. The function of cutin is to make the leaf less permeable to water, so it actually traps water in. If you didn't have cutin, the leaf would dry down very quickly, so the cutin actually helps control water movement through the outside of the leaf. This is a scanning electron micrograph of wax on the surface of a leaf. And you see some different kinds of structures in here, but again, they are long chain polymerized hydrocarbons. This shows you a leaf with a glossy surface, and this glossy surface is due to cutin and wax. They say if you have houseplants like this and you want to clean them, you take a little bit of milk and a piece of cotton and you clean the outside of it, and it cleans it up very well because milk has hydrocarbons in it. Here's candle wax. This comes from petroleum, but the petroleum material actually has these long chain polymerized hydrocarbons in there that are very stable and are waxes. And that's what your candle looks like when it burns. So we know it has energy in it. Put the match to it, you can burn it, and you get this nice little heated flame out of it. The other types of material we've got here, we talk about Suberin, and Suberin is like cute, but Suberin occurs on below ground structures. If you've ever seen a potato that has been cut, when it dries, it gets kind of a waxy-like material on the outside of it, and that waxy-like material is Suberin, and the function of the Suberin is, again, to keep water from moving in and out of that particular plant material. In the end, when we're dealing with lipids, a lot of different kinds of lipids, they all have the similarity in that they go back to hydrocarbons that are there for energy storage, but they are also hydrophobic, meaning that they do not like water.